Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. In today's lecture, we are going to discuss the polymerase changes and the half form is the PCR or the short form is the PCR. This is also called the molecular photocopy because in this process, we can prepare the identical copies of the target DNA, thus multiply the available DNA and it gives us enough sample to study or to test multiple types. We can study or we can design the multiple experiments to get the multiple objectives, which is not possible if we cannot or we are not able to amplify the amount of the samples, DNA samples available to us. It is only possible because of the PCR. So before going to the details of the PCR, we should revisit the history of the PCR. It was invented in 1983 by Kerry Mullis, who is a technician at the Citrus Corporation. And the first paper was published in 1985 by the Citrus Corporation based on the PCR. In 1986, the TAC polymerase, the enzyme that is purified from the Thermophilus aquaticus, it was purified and used for the PCR. In 1988, whatever is going on manually in the 3 incubation steps, it was given a machine shape and we got the automated thermocyclone that we call it the PCR machine today. In 1989, the TAC polymer that gives a lot of efficiency to the PCR or the PCR is possible. It was declared as the molecule of the year by the Science magazine. In 1998, the fluorescent dye used in the PCR, it gives the birth of the real-time PCR that is used for the testing of the corona in this year 1920. And after that, in 1991, the virus was identified as a diagnosed using the PCR machine. Considering the importance of the PCR, the revolution it has done in the biological research and development in the health science, Kerry Mullis shared the Nobel Prize in 1993. The PCR or the conditions of the PCR, it is of 3 steps: denaturation, annealing, and the extension. So for the denaturation, the temperature is between 90 to 95 degrees Celsius. It is written 94 here, but we usually use the 95 or the 90. In this process, whatever DNA template we have, that we linearized, denaturation, and it is separated. Then the annealing, in which we see the forward primer, the reverse primer, they hybridize with the template DNA. And this is the extension. It is usually the 72 degrees Celsius for the various DNA polymerase using the Thermophilus aquaticus. And this enzyme, they usually amplify 1 kb of the DNA in around 0. In 30 seconds to 1 minute. So depending on the how much amplicon we have, what is the size of the amplicon, the extension time it varies. We usually put between the 30 seconds to 1 minute. This process, the denaturation, the annealing, and the extension, it repeats with time in the PCR. So this is the cycle 1, the 3 steps. It is done only one. From the 1 copy, you get the 2 copies. In the cycle 2, the 2 copies both act as the template, so we can get the 4 copies. In the cycle 3, we get the 8 copies. Similarly, in case of the n number of the cycle, the total number of copies is 2 to the power n. So the overall principle of the PCR, here we have the DNA 1 copy. We make several copies. So can we do the several tests. What are the components we use in the PCR? The magnesium chloride. What is the importance? We will discuss in the following slide. Buffer. We know that buffer is a solution in which if there is an addition of the small amount of the acid or the bases are done, then the pH does not change. Or it is a solution that resists the change in the pH when a specific quantity of the acids or bases added to it. So it is very important for the activity of the enzyme. Then the D and TP, deoxynucleotide triphosphate, these are the building blocks of the DNA. So we provide the building block. We need the primer because it will provide a 3' OH and DNA polymer is the enzyme that will make the polymerization and the template DNA. So what is the importance of magnesium in this case? This is the DNA polymer. It is the hand shapes. This is the palm. This is the thumb finger in the palm. So here the addition of the new nucleotide takes place. So there is the importance of a yeah, double ion, double positive and the magnesium. So the presence of the magnesium here, it is providing the electrostatic interactions. It is donating the two electron. The two positive means it is accepting the two electron from the two oxygen. Like this, it is forming the bond and it is helping in the polymerization process. So the bivalent cation, there are the various bivalent cation, there are the various roles, like the Fe2 plus iron, every one of us know that it has very role in the hemoglobin for the oxygen carrying. Similarly, the PDG domain zinc bivalent cation has the role. So in case of the DNA polymer, the bivalent cation is the magnesium. That's why we need to supplement magnesium considering the importance. Then what is the importance of the PCR? It is highly specific, highly efficient, and it is verified, or there is a fidelity. So there is no error. That's what when the size is less than 1.5 kb, which is the taq polymerase. When the size is more than 1.5 kb up to the 3.5 kb, we use the PFU polymer. So depending on the size of the amplicon, we change the DNA polymer. That is based on the fidelity, means how much it can polymerize without any error. These are the few examples reported in the early days of the PCR, the actually human leukocyte antigen typing, the HIV-1 identification of the diagnosis. Factor V lead-in, that is their role in it is a blood clotting factor. These are the two species, chlamydia, trachomatis, and the Neisseria, gonorrhea, and various forensic testing and the many others. This is in the early period of the PCR. Nowadays, molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organism, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. These are the various role of the PCR, that is for the molecular identification. Then bioinformatics, genomic cloning, human genome project, these are the role of the PCR in the sequencing. Similarly, in the genetic engineering, it can be used for the site-directed mutagenesis and the gene expression studies. So a machine, depending on what primer we use, depending on the size of the amplicon we use, we can use this PCR technology for getting all these objectives. This is the classical PCR. Next, what is real-time PCR? How it is different from the classical PCR? So usually there are the two types of the dyes are used, fluorescent dye in the real-time PCR. One is the SYBR-Green dye, another is the TaqMan fluorescent detection. So in case of the SYBR-Green, once the duplex DNA is prepared, means one cycle, that is the one duplex DNA is formed. In that case, when the SYBR-Green is bound or the intercalate, the DNA double stranded, then it fluoros. So we see the signal. So if the lot of copies of the DNA, we see lot of signals. So this is the basis of using the SYBR-Green that usually intercalate in a double stranded DNA and it gives it fluorescence, thus we get the signal. The another one is the TaqMan, fluorescence detection process. In that case, along with the forward primer and the reverse primer, we use a probe. The probe, the two ends, one side it has a reporter with one color, another is a quencher. So as long as the probe is intact, we don't see the fluorescence. Once we see the amplicon is formed here, the reporter, it is separated from the quencher because probe is removed or it is degraded. So we see the fluorescence. So more copies, the more probe will be degraded. So that reporter and the quencher will be separated and we see the more fluorescence. It is more accurate and it gives you us more accurate data than the SYBR-Green methods. And this TaqMan is used for the multiplexing that we will discuss in one of the following slides. So what is the advantage of this PCR? One of the characters of the primer designing that primer should not dimerize. So in case of the real-time PCR, this peak provides us information that whether the primer is good or not. If there is a primer damage, means we have to design the new primer. That we cannot get in case of the classical PCR. So it is validating our primers also. And it is giving us an opportunity to design the new primer. That gives us a more accurate data. Then this is the amplification plot. So in the early period of this is the relative fluorescence unit. This is also called the delta Rn. So R for the relative. So if it is a CT or the early cycle, you see the amplification, that means we have lot of template. If we see the late amplification, then we have less template based on this it works. Then what are the various parts of this S-shaped curve? So this is the relative fluorescence unit, this is the cycles, and here this we make this threshold. So once the signal is visible, it crosses the threshold, we call it the cycle, at which we can see the signal. And this CT is used for the identification, quantification, or comparison of the template amount of the DNA. This is the exponential phase and this is the plateau. So this is another importance of the real-time PCR that we can prepare the standard curve. This is the amount of the template DNA we exactly know, and this amount of the template DNA at which time it is giving us the signal, the CT values. So if the amount of the template is less, the CT is 35. If the amount of template is high, the CT is low. And from this, we can get the unknown quantity of the template DNA that we'll discuss in the exact data. So this is the SARS-CoV-2, how it was identified. This is the genome of the SARS-CoV-2. It is almost the 30 kb. Okay, and here we have up to 10 open reading frames. This is the RNA-dependent RNA polymerase. This is the envelope gene. So these two and one of the reporter from the human that is used as the internal control. So when each of the genes are studied separately in the three worlds, it is called the simplex. That is being used in case of the SYBR-Green and in the old classical way of doing the real-time PCR also. This is the duplex, means each sample has the reference gene along with the viral gene. RNA-dependent RNA polymerase and the human gene. How we can separate these two based on the two colors. One is the green, another is the hex color. Or similarly, or the VIC color. So we have the four types of the color in one machine, one sample. You can study the four genes at one time in case of the PCR. So this is the duplex. In this case, we can see the three genes all the together. How we can separate the three genes depending on the three colors or the three fluorescence. Better to say. What is the exact data? This is the RDRP gene. Here we can see there is a 10 virus particle in the sample. You can detect 10 literally. So this is 10, this is 100, this is 1000, this is 10,000, this is 1 lakh. So it is more specific. And this E gene, it is slightly less specific than the RDRP. That is, the primers are less specific, or the threshold is higher in case of the E gene. So we can see up to the 1000 particle, here we can see up to the 10 particles. This is based on the viral load. Thank you.